Welcome students. I'm Dr. Sima Desai Fernandez from St. Xavier's College, Mapsa, Goa. This program is for the Bachelor of Science first year students, subject Botany, semester 2, course code BOC 102, course title Biodiversity 2. Title of the unit Pteridophytes, module name Sexual Reproduction of Selaginella. Outline. The module will cover sexual reproduction in Selaginella. Learning outcomes. It explains the sexual reproduction in Selaginella. This is the picture how the Selaginella plant looks like. Introduction. Selaginella is heterosporous. There are two types of asexual spores, microspores in microsporangium and megaspores in megasporangia. The presence of two different sized spores in the same species is known as heterospory. The sexual reproduction in Selaginella takes place by gametes, eggs and sperms. Sporophytes produce asexual spores which germinate to produce a small sexual plant, that is gametophyte, and it is called as prothelium. Spores, there are two types of spores, microspores. Let us see about the microspores. The microspores are small in size. It is 0.015 to 0.06 millimeters in diameter, spherical or round in shape and double-layered structures. The outer wall is thick and known as exospore, also called as exine, while inner wall is thin and is called endospore, also called as intine. The haploid microspores on germination give rise to male protheli or male gametophyte or microgametophyte. Let us see about the megaspores. The megaspores are much larger than microspores, 1.5 to 5 millimeter in diameter, tetrahedral in shape and show triradiate ridge. The megaspore has three wall layers, namely exospore, mesospore and endospore. On germination, the haploid megaspore develops into female protheli or female gametophyte, also called as megagametophyte. Let us see the gametophytic generation. The development of male and female gametophytes, that is protheli, takes place from the haploid microspores and megaspores respectively. That is, microspores and megaspores are the unit of male and female gametophytes respectively. The development of microspores and megaspores generally starts inside their respective sporangia. Therefore, the spores are shared at multicellular stage. Gametophytes produce gametes, eggs and sperms and fuse, giving rise to the sporophyte generation. In Selaginella, male and female protheli are formed and confined within the spore walls. The spores are formed in sporangia, which are one-celled, subglobose and solitary. Male gametophyte develops within the microspore wall. Male gametophyte develops antheridia, which produces anthrozoids. The anthrozoids are free-swimming type, spirally coiled and biflagellate. Megaspores are large in size, 1 to 4 numbers. They germinates into female gametophyte. Female gametophyte produces archegonia, which produces eggs. These reproductive structures after fertilization produces zygote, which again on germination give rise to a sporophytic plant which is diploid. The young sporophyte remains attached to the megaspore for some time, later falls to the ground and form roots and become an independent plant. In this way, the sporophytic and gametophytic generations alternate with each other, although the sporophytic phase is dominant over the gametophytic phase. This is the structure of the mature archegonia, which is showing the neck cells. Below the neck cell, there is a venter and it covers one egg inside the archegonia. The archegonia is a short flask-shaped structure. This is a structure which is showing a flask shape and embedded in the female gametophytic tissue. Only the upper tier, that is here, only the upper tier of the neck cells project out. Each archegonia consists of a short neck of 2 tiers of 4 cells each and a broad venter. This is the picture which shows the broad venter. The 4 cells of the upper tier of neck function as cover cells. The neck encloses a single neck canal cell and venter consists of a ventral canal cell and an egg. There is no definite wall of venter. At maturity, the neck canal cell and the ventral canal cell disorganize and absorb water, which creates a pressure to separate apart the cover cells, through which the anthrozoids enter the archegonia and reach the egg. Here the rhizoid, this is the picture of the female gametophyte which is showing the rhizoids. This is the archegonia which is a flask shaped and there is embryo here and this is the endosperm. Fertilization. The mature archegonia are embedded in the gametophytic tissue. The ventral canal cell and the neck canal cell degenerate, leaving only the egg. Fertilization may take place when the megagametophytes are on the soil or when they are still within the megasporangia. Water is necessary to carry out the process of fertilization. The swimming anthrozoids reach the egg through the neck of archegonia and the nucleus of anthrozoid fuses with the egg nucleus, thus forming a zygote nucleus. The fertilized egg secretes a wall around it, forming a diploid structure known as zygote or oospore. Thus, the gametophytic generation ends and the initial stage of new sporophytic generation is formed. In some species, example, Selaginella intermedia, the egg develops into embryo without fertilization. This phenomenon is known as parthenogenesis. Summary: the sexual reproduction in Selaginella takes place by gamete formation, that is sperms and the egg. The haploid spores are produced in sporangium. Male gametophyte develops within the microspore wall, produces antheridia, which bears anthrozoids. The anthrozoids are free-swimming type, spirally coiled and biflagellate. Megaspores are large compared to microspores and germinates into female gametophyte, which develops archegonia, which produces eggs. Anthrozoids fertilizes the eggs and the new sporophytic plant is developed. These reproductive structures after fertilization produces zygote which is diploid, which again on germination give rise to a new sporophytic plant which is diploid. These are the references. Thank you.